Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is NanoGBS, a miniaturized procedure for genotyping by sequencing library preparation, and our sponsor is Beckman Coulter Life Sciences. Our panelist for today is Dr. Davoud Torkamaneh, Assistant Professor at Université Laval. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. Click to the tray at the bottom of your window. You'll see a series of widgets to enhance your webinar experience. And with that, I'll hand it over to Dr. Torkamaneh. Please go ahead.
Thank you, Kirsty, and I'm happy to be here with you. And today, as Kirsty said, I'm going to talk to you about a NanoGBS. It's a miniaturized procedure for genotyping by sequencing library preparation. My lab is located in the beautiful Quebec City in the eastern side of Canada, and it's located in the Institute for System and Integrative Biology at Laval University. It's somehow the most largest French-speaking university in North America. Without any further ado, I'm going to show you what I'm going to present you today. At first, I will discuss a little bit of what is a genetic variation, what is a genotyping by sequencing, and why it is a good fit for us. Then I will very, very briefly to touch on genomic selection and why we need a high-throughput, low-cost genotyping platform. Then discuss the acoustic liquid handlers and how we have implemented NanoGBS. Then I will show you some strengths and limitations of this method, and I will finish with some concluding remarks. Genetic variations are the difference in the DNA sequence among individuals within a population. As an example, here you can see we have two samples. They are mostly identical in all across the genome, in every basis of the DNA, and in some places they are differing at a nucleotide level. When they are different at the nucleotide level, there are nucleotide variants. The most common one is SNP, or single nucleotide polymorphism. We have the second type of the variation in the genetic level called structural variants. They are mostly 50 base pair larger, and they can be seen as large deletion, insertion, translocation, inversion, duplication, or copy number variation. Today, we are mostly focusing on nucleotide variants, specifically again the SNPs or SNPs, where they are very frequent across the genome. As an example, we can find at least one SNP in every 1,000 base pair in the genome. They have a viral role, means they can affect the phenotypic makeup of the individual. As they can affect the phenotype, so we can use them as a marker. So we can associate these genetic variants with the phenotype of interest and develop a molecular marker. So as genetic variation can explain the difference, so they can be used as a molecular marker for diagnosis, for the screening, and also for the breeding. The first step is to detect or discover this genetic variation, or genotyping of the SNPs. What we mean to determine the SNP loci on a whole genome scale or within a genomic region of interest. There is different type of the technologies or methods that we can use to genotype the SNPs. The most common ones are SNP microarray, genotyping by sequencing, or whole genome sequencing, where we sequence the entire genome of the individuals. Each of these methods, they have their own merits and demerits, and I try to summarize some of them in this table. For microarray, if you're familiar with that, we need some pre-investment. Means we have to sequence the population and find the variation, genetic variants within this population, and try to develop a microarray based on this variation. And they are mostly gene-based. Their cost is around 100 to 150 bucks per sample, and their limitation is, their pre-investment, distribution, and also the cost. The second approach is GBS, or genotyping by sequencing, where there is no need for pre-investment. It's a targeted method, means we target a specific regions of the genome. The cost per sample is around $20 to $30 per sample, and the limitation is data analysis and the missing data. At the end, we have the whole genome sequencing approach, where again there is no need for pre-investment. We sequence the entire genome. It cost differs between 500 to 5,000 based on the genome size and the species. And the limitation is, again, the cost, data analysis, and sometimes it's not necessary to. Genotyping by sequencing approach, which has been developed in 2011 and then improved by my colleague in our lab, Sona et al. In 2013, it simply uses the restriction enzymes and cuts the DNA in a small fragment, and then we sequence the specific regions of the genome that has been cut with the restriction enzyme, and we sequence these genomic regions and try to find the genetic variation that exists within the individuals. How it works in the lab. We start with the DNA plates, then we add the restriction digestion mix into the plate, then ligation mix, the adapters that are sample-specific barcodes and also universal adapters for the sequencing. When each sample have their own specific barcodes, we can pool them all together. We run a site selection to select only sequenceable fragments. Then we run a PCR amplification to amplify the fragments, then quality control and sequencing. After sequencing, we have the data analysis, and data derived from GBS protocol has been used for different type of studies in genetic experiments, for like association analysis, like wide association analysis, for QTL mapping, for genetic diversity analysis, and also for breeding purposes like genomic selection. Here I'm trying to very briefly to present what we mean by genomic selection. But before that, I explain you what we do in the conventional breeding at the left side of the figure. We cross through parents. Parents normally generate a population. Then we select them for multiple generations. Then we try to phenotype all these samples and select based on their phenotypic data. In this setup of the breeding, normally we have thousands to millions of individuals that has to be phenotyped and run through selection. But in genomic selection, what we do, at first we start with a training population. It's a diverse population that includes also the parental lines. We genotype and phenotype them extensively and use this data to train a statistical model. And use these models after a few generations of the plants to only genotype the progeny, then feed this data into the model to predict their phenotypic output. So we try to eliminate the step of the phenotyping and run the phenotyping and at the end for the selected genotype. So this approach requires fast and cost-effective genotyping platform. As I said a few seconds ago, we need to genotype thousands and sometimes to millions of the individuals. So we need a cost-effective genotyping platform. But the question is how we can decrease the genotyping cost. In general, the genotyping cost is composed of three components: the library preparation cost, sequencing cost, and data analysis cost. I'm sure you are all aware of the sharp decrease of the sequencing cost in the last few years. And we can say that now the sequencing is not a big problem because there is a different type of the technologies that can generate a large amount of the sequencing data for a very low cost. Also, in the last few years, we have done a lot of progress and improvement in the data analysis, the bioinformatic data analysis, where we can now very quickly, fast, and cost-effectively process the sequencing data. So we have done a lot of progress in two sections, sequencing and data analysis. But still, the library preparation is the very similar way that been doing for the last 10 years. We haven't done much improvement in that component to reduce the cost. I want to go into the details of the library preparation cost or the materials that we use for the GBS library preparation. In general, we use 10 microliter of the DNA, then 20 microliter of restriction enzyme digestion mix, 15 microliter of the ligation mix, 5 microliter of the adapters. And we have a mix of 50 microliters. But if you remember, we pool the samples together. And during pooling, we use only 5 microliter of each sample. Means we prepare libraries for 50 microliters and use only 5 microliters. Means we are having 45 microliters extra. But how we can decrease this, the use of the materials? The problem in the lab was we couldn't go under 5 microliters based on the pipetting system and the errors that can be happened during the pipetting. Since we found the acoustic liquid handlers. How the eco liquid handler works? Very simply and briefly, we can say we have a source plate where we have all the materials that we want to transfer. Then we have our destination plates. The destination plates will flip the turn around, will turn, and using the sound energy system, we try to send the droplets of the materials from the source plate to the destination plate. With this method, we can transfer the droplets of 25 nanoliters, very small droplets, with a very high accuracy and precision. It's a very fast and flexible method. We can transfer up to 6 microliter per second from any well from the source plate to any well in a destination plate. It's a very precise, accurate, and fast liquid transfer using sound energy. For us, the reason why to use the Eco liquid handler was we can reduce the volumes and therefore the cost for the library preparation. The technology is tipless and therefore no use of the plastic. Again, we can decrease the cost. This technology can help us to reduce the potential cross-contamination that we have with the pipetting, the manual pipetting system. We can adjust the transfer parameters in real time for variety of fluids. Various samples and reagents at different concentration can be transferred using this machine without any user calibration. Finally, DNA, RNA, enzyme, and any master mix in blue storage solution can be used to be transferred. So the take-home message is reduced reaction volumes automation is a key to cost and time saving in mid to high throughput application. So using Eco technology, we tried to develop the NanoGBS protocol. As you can see in the table at the left, you can remember the volumes for the StdGBS approach, StdGBS, and then for the NanoGBS, we try to reduce the volumes by tenfold. So means we use only 1 microliter of DNA, 2 microliter of enzymes, 1/2 microliter of ligation mix, and only 0.5 microliter of adapters. And our final reaction was 5 microliter. And we used the totality of this reaction for the pooling. Means we had 0 waste in this case. In the picture, I just try to show you the different steps in the StdGBS where we use the manual pipetting to transfer all these materials, and through the NanoGBS we use only Eco machine to do all this liquid transfer. So in the NanoGBS methods, we could save 90% in reagent usage. But the question was, is the quality of the genotype stay the same when we reduce the reagent volume? So we ran the NanoGBS and StdGBS on 96 soybean samples, for which we have the whole genome sequencing data, a very solid data set to be able to compare the results. We prepared the libraries around the sequencing, and then we measured the quality of library, sequencing, and genotyping. In the figure at the top, we have the libraries developed using NanoGBS approach and also a StdGBS approach. You can see the libraries, they are identical in both approach. We have only less materials in NanoGBS as we use the less material at the beginning. Then we run the sequencing with the same sequencing technology, same protocol for both libraries. And then we measure the number of the reads per sample in different protocols. As you can see, The number of derivations that we generated for each sample using NanoGBS or StdGBS remains very similar. And even the uniformity that was higher in NanoGBS protocol, and we think that's because of the precise transfer of the liquid during library preparation. But the most important and significant result was the genotyping part, the SNP calling. We found more than 96% overlap between the SNP data derived from the NanoGBS and StdGBS for the same samples. And most importantly, we found 100% of the agreement between these two methods. This means that with reducing the volumes of the materials for the library preparation, we can still generate exactly the same high-quality genotype. Finally, we tried to use the NanoGBS library and increase the multiplexing level. Instead of doing only with 96 samples, we increased the number of samples to 384 and 68 samples together and sequence them and try to estimate the cost. We try to have all the costs related to the genotyping from the reagent, labor, and sequencing. And we found that with the increased multiplexing and using NanoGBS library preparation method, we can save up to 72% of the cost. On the other hand, we save not only the money. Using the NanoGBS method, we reduce the handling time by 75% compared to StdGBS. Again, the saving in the labor cost. Here you might say that we've been able to reach our goal of decreasing the genotyping cost. But that's not the end of the story. If you remember the different steps of the library preparation for the GBS, in every step we were using the manual pipetting with the pipette, and in each step we were using a high quantity of the plastic tips. However, when we do the NanoGBS, we are not using any plastic tips for the transfer, as the technology is a tipless transfer. Why this is important? The Genomic Lab is one of the source of the production of the plastics because we use tons of the tips for the manual pipetting. And the sad part is that most of these plastics that we use in the genomics labs, they are not recyclable. So based on the different studies, now we can say the genomics labs and the bioscience labs, in general, they are one of the sources of the producing the plastics in the world. So with NanoGBS, we can say that we can save and reduce the use of the plastic in genomics lab. As a concrete example, we try to measure the quantity of the plastic that we use through StdGBS method, where for only one plate of 96 plates, we use around 63 grams of the recyclable plastic, 59 grams of the non-recyclable plastic, and 122 grams in total for only one plate. But using the NanoGBS, we reduce the recyclable plate to 0. Literally, we are not using any plastics, recyclable plastic. And we use only 14 grams of the non-recyclable plastic. They are the plate, the source, and the destination place that we use in the machine. And in total, we reduced the use of the plastic by 89%. So we can say this approach is very eco-friendly and can be very useful to reduce the use of the plastics in the genomics labs. But finally, we have some health issues related to manual pipetting. You may say that it's not a big deal, yes, but if you are using just one plate, that's okay. But in the lab like ours, when we are pipetting 50 to 100,000 reactions per year, the quantity of the work is quite important. This can result in a repetitive strain injury, work-related upper limb disorders. That's very common for the people that are pipetting every day, and also musculoskeletal disorder because of bad positioning in the lab. But now in lab we have the echo used for all the transferring works. And I think our personnel in the labs, they are very happy with this machine too. Finally, as a concluding remarks, I can tell you that we have developed an ultra-low-cost genotyping method, NanoGBS. We could reduce the genotyping cost by 72%. That's very impressive and can help the people to use more genotyping methods. We developed an eco-friendly library preparation protocol that can help to reduce 89% of the use of the plastic. Most importantly, with the reduction in the volumes, we still got the high-quality genotypes. And even we increase the uniformity of the genotype and decrease the missing data. And this protocol can be used in any species for the library preparation or any different NGS library preparation. But the downside is that liquid transfer technology, the eco machine, remains costly, and for some labs can be very expensive to purchase such machine. But in our case, it was very it can return its cost in only a few years based on the savings that we can do both on time and the materials. Our future direction is to automatize more our library preparation protocols. We might integrate the different biomech liquid handlers and robotic system to be able to automatize our processing more and again to save time during the work. Thank you all for your attention, and I would be happy to take any questions.
Thank you, Dr. Torkamaneh. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. And joining us for the Q&A today is Sohaila Beck, Commercial Product Manager for Genomics at Beckman Coulter Life Sciences. All right, let's start with our questions. Okay, so Davoud, our first question, could you tell us if you saw any inherent advantages of using Ion Torrent compared to Illumina or other sequencers in your lab?
Thank you. That's an interesting comment because we had the Ion Torrent machine in the lab. We mostly use Ion Torrent, and the reason why we were using Ion Proton with the Ion Torrent technology was during the genomic selection work, the timing is really important for us, and sometimes we need to sequence quickly every sample that we receive. In the case of the Illumina, we had to send out outside in the Genotyping sequencing platform, and it might take a few weeks to get the results back. But with the Iron Torrent, we were doing in-house, and that was the reason why we were mostly using the Iron Proton technology in the lab, because of its rapidity to generate the sequencing results.
And you mentioned missing genotypes in GBS. Could you elaborate a little bit on that and how the NanoGBS could overcome this issue?
That's a very interesting situation, because in GBS, in any approaches, the RAD, GBS, the different type of the genotyping or sequencing methods that we use, restriction enzyme or fragmentation, and then we pool the samples together. By nature, we will not be able to capture all these fragments to sequence. So this can generate missing data in some genotypes. It's not possible to get to reach the 100% presence of all the genotypes in these approaches. But the one part is really important. We can at least have a uniform type of missing data. Sometimes can happen that we use some samples 100% are missing because of the DNA, the process, everything, and the manual pipetting can help to resolve this. But with the NanoGBS, with using this with using the precise liquid transfer, we could at least uniform to have a uniform samples for everything except the DNA, the input DNA. If the DNA was also the same quantity and quality as the others, the NanoGBS helps us to create more uniform libraries. More uniform libraries means we are going to have less missing data issues. And we've seen that in our case, because it was just a test. Now we are optimizing the protocol. We've seen a quite interesting reduction in terms of the missing data when we were using this NanoGBS approach.
Have you found that the miniaturized volume changes the kinetics of the enzymatic reaction at all?
Not in my experience. We've done that and it worked perfectly. Actually, we've fragmented exactly the similar way that we were doing with the actual volume. In our case, we haven't seen any problem with that.
And has the miniaturized liquid handler affected DNA fragment length at all, which might be relevant for nanopore sequencing?
In our case, we are saying NanoGBS, actually we need the fragmented DNA. DNA is more fragmented, the better the library will be. We haven't tested for the nanopore sequencing, might have an impact on that, but when we are talking of the DNA fragments or DNA in general, they are very tiny. Even I think we can be able to transfer the actual DNA with the one droplet or a few droplets without fragmenting or cutting the DNA in small pieces. But in our case, we haven't had much interest on that because we were working with GBS high throughput, high volume samples, thousands of the samples. But with nanopore, we do nanopore sequencing, but our number of the samples is very low. Just few samples, tens of samples. and we haven't tested the echo or droplet transfer to see what will happen with the DNA of large fragments.
Great, thank you, Davoud. Sohaila, I have a couple of questions for you. Could you tell us what the maximum volume is that the echo can dispense?
That's a great question. Echo dispenses Echo 525 25 nanoliter droplets really fast, a couple hundreds of them in a second. And we have customers going up to 5 or 10 microliter. We recommend keeping the volumes low for the sake of the speed, but because of the accuracy and precision they get, some people have been doing 96 well plates on 5 and 10 microliter. But again, I say my recommendation would be keeping it below 5 microliter.
And for echo dispensing, should the source and destination plates always be flat-bottomed?
That's another great question. The source plate should be an echo-qualified source plate and a flat bottom because of the way ultrasounds transfer through the plate to focus on the meniscus of the liquid inside the source well. However, the destination plate can be any SDS standard 96 or 384 well plate, as long as they either have adapters to hold the plate if it's semi-skirted, or if it is fully skirted, we can work with it.
Great, thank you. So Davoud, back to you. Are miniaturized libraries more uniform? Have you found that in your research?
Yes, with the NanoGBS, we had more uniform libraries in terms of the volume, and at the end with the sequencing, we've seen that the libraries prepared with the NanoGBS approach, they were more uniform than the standard or the conventional libraries.
And would someone need to make major changes in their lab workflows in order to use the echo?
Not actually. In our case, we just, as I said, we are working with the high number samples. Before echo, we were doing everything in 96 plex, and our PCR machine, everything were set up with 96 well plates lead. But now with the echo, the only thing we have changed that was only getting new PCR machine with 384 well plate leads. Other than that, everything they were the same process we are doing, just we are using echo.
And how do you control the evaporation when working at the nanoliter level?
That's a very interesting question, because it was we could go even lower than 1 microliter to go to 25 nanoliter of the liquid. We've tested a few we've run a few tests with the very small volumes, and we've seen that if we go around 1 microliter, we are 100% safe to have the liquid there. For DNA, actually, it doesn't have any impact for us, because DNA is it's just mixed in the liquid, and even with the evaporation, DNA remains in the plate. There wasn't that problem. And another thing we are doing, we're fast tracking the steps. We are not just transferring and waiting. Very quickly, we pass the different steps. And the very technical point was using the 384 well plates, because the form of the plates is different from the 96 wells, and it reduces the evaporation of the materials.
Thank you, Davoud. Sohaila, sure. Should a user worry about volumes falling down when the destination plate is held upside down, or is that not an issue?
That's a good question. The volumes stay in the destination plate by surface tension. Obviously, that depends on the liquid type and the coating, but let's say I have water in a 96 well PCR plate. I have been able to go to 20 microliter upside down, no problem, because the surface tension holds the volume upside down, no problem. And that's how ECHO has been used historically. Always the destination plate has been upside down, and it has been no problem.
And after the addition of the sample and the reaction PCR master mix, does it require mixing before the PCR is actually done?
That's a great question. So we thought that at the beginning, we need to mix it because in larger volumes, we traditionally mix the volumes. But because we are reducing the reaction volume, it actually takes care of mixing by itself. And once you insert it into a turbocycler, that heat transfer also mixes it more. If there was some bead that needed to be mixed or something like that, I understand we can seal it and vortex it. But for I let Davoud actually answer this one too. But for miniaturizing the reaction volumes, because the volumes are low, when I was working with ECHO myself, I didn't see a need to mix because the volumes are small, and Kinetic just takes care of it. Yeah.
Yeah, Davoud, any?
Davoud, do you want to add here?
Yeah. Could you please repeat the original question for me again?
Sure. So after the addition of the sample and the reaction PCR master mix, have you found that there's any need to mix before doing PCR?
No, we don't do any mixing after PCR. I think it's very simple, physics and the chemistry. I think when you have a higher volume and transfer from the smaller to the higher, they will mix very well, and we haven't seen any problem with that.
Okay, great. Sohaila, are there any devices that you would say are similar to the ECHO?
ECHO remains to be the only acoustic liquid handling technology that uses sound energy and no tips to transfer the liquid from source plate to destination. There are other low volume liquid handlers, but there is nothing like ECHO that can transfer from any well in the source to any well in the destination in under a second, because it's not using any mechanical, tip or plastic to move the drops. I can say there are other nanoliter, microliter liquid handlers, but ECHO is the only acoustic one that, because we are not using tips or plastics, we don't have any liquid absorbing around the plastic and affecting the accuracy and precision. The key stays in transferring drops without any touching, and therefore achieving that high accuracy and precision.
Great, thank you very much, Sohaila. And that was the last question that I show in the queue, so I'm going to wrap it up there. We'd like to thank Davoud Torkamaneh, Sohaila Beck, and our sponsor, Beckman Coulter Life Sciences. If we didn't have time to get to your question, we will try to follow up with our experts later on. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.